Distinguished judges and teachers, my name is Sophia Zeng. I'm from Tsinghua International School. I'm very excited to present my research on the impact of high caffeine intake on adolescent brain development. Why this topic? Well, the public health implications are very real and significant, and yet very little is known from a scientific perspective. What have I learned? For adults, caffeine can be both good and bad. However, for adolescents, high caffeine intake adversely affects the development of the central nervous system. In terms of research methods, I learned quite a bit of multi-level experiments in bioinformatic analysis through this study. Let me begin by providing some context for my study. Caffeine intake is rising among adolescents through energy drinks, coffee, and caffeinated beverages. According to some recent statistics, 83% of adolescents consume caffeine regularly. Exactly how excessive caffeine exposure might affect the long-term trajectory of brain development and function remains a mystery at the molecular, cellular, and behavioral levels. Recent literature suggests that epigenetic mechanisms, particularly histone modifications, are a major factor. We hypothesized that excessive caffeine exposure during neurodevelopment disrupts epigenetic regulation, leading to suppression of neurodevelopmental gene programs, impaired neuronal maturation, and subsequent behavior dysregulation. To test this hypothesis, we've employed an integrated bioinformatics plus experimental approach. We use omics analysis to identify caffeine-responsive genes and epigenetic modifications. In our experimental design, primary culture neurons were measured to assess different effects on neuronal morphology, and zebrafish embryos and larvae were observed to examine developmental and behavioral consequences. The first set of results is concerned with transcriptomic findings. In the volcano plot shown in the top left figure, genes in red are significantly upregulated following caffeine exposure, while genes in blue are significantly downregulated. This plot clearly demonstrates that caffeine induces widespread transcriptional changes. The bar plot at the bottom shows the top 20 upregulated and downregulated genes. As we can see, caffeine exposure causes global disruption of gene expression profiles, many of which are involved in fundamental neurodevelopmental processes. Further pathway enrichment analysis confirmed these findings. At the cellular level, the figure on the left shows MAP2 immunofluorescence images of primary culture neurons following caffeine treatment. We know reduction in neurite growth. Shoal analysis, on the other hand, shown in the figure on the right, demonstrated dose-dependent reductions in neurite complexity at distances of 20 to 80 micrometers from the cell soma. These reductions are all statistically significant. Furthermore, in our zebrafish model, qPCR analysis revealed significant suppression of critical neurodevelopmental and neurotrophic genes. At the organismal level, the observed molecular and cellular alterations translate into profound behavioral dysregulation. In zebrafish larvae exposed to caffeine, locomotor activity assays revealed hyperactivity with much heightened anxiety-like behavior and impaired cognitive function. In conclusion, our research contributes to the critically needed exploration of the neurodevelopmental impact of excessive caffeine exposure. We found a range of adverse effects at the epigenetic, cellular, and behavioral levels. Our research findings can help inform public health policies and educational initiatives aimed at protecting adolescent brain health. Thank you.